Li- Hello, guys. Hope that you are good. Today, I'm going to talk about activation of macrophages. In the previous lectures, we learned about macrophages that these cells are present right from the early, embryonic development during the gestation. These cells are present in the embryo or the fetus, and these cells uh, are, in the embryo, they are made out of the yolk sac or the liver progenitor cells, and those macrophages which are produced during the embryonic development or of the embryonic origin later become fixed macrophages in different tissues. They are also called resident macrophages. And after the fetus is born, uh, the bone marrow is activated, and the bone marrow starts making these different types of white blood cells and the red blood cells, and the macrophages in the form of monocytes are formed from the stem cells, which enter the blood, and from the blood, these cells can later enter into different types of tissues and become, macrophages. We again, the monocyte-derived macrophages are also called monocyte-derived macrophages. These are actually coming from, the bone marrow or the blood tissue. So these uh, macrophages again can join the tissue-based macrophages which are already there in different tissues, and they can, help out with the function of the tissue macrophages. Now, these are magical cells. These macrophages are not only involved in immunity, as I told you in the previous lecture, that it's not only the human defense system that the macrophages are important for. The macrophages are playing important roles in other processes like development and metabolism. They are involved in all these different types of activity, and they are the most adaptable cells in our body. And uh, there are kind of unique cells that can be activated in two different ways. One is called the classical way, and the other is the alternative way. Or more simply, we can put it like this: that the classical way is pro-inflammatory, that it promotes inflammation, and the other alternate pathway is anti-inflammatory. So um, it's doing two contrasting things at the same time. And these two types of activation, whether pro-inflammatory or anti-inflammatory, um, depends upon the situation or the contact partners of the macrophages, that how the macrophages come in contact with different things that can activate it or switch it on differently. So inflammatory is a protective response of your body, and it involves a lot of different types of your immune cells and the products in the form of different cytokines that these cells actually are making. And the purpose of inflammation is to resolve the infection, or if there is any kind of other problem, some kind of necrosis or something which is going on inside the body. So the purpose of that response in the form of activation of different immune cells is to just clear off that infection or the debris, or whatever is happening there, some pathology. And the purpose of this kind of activation, the anti-inflammatory activation of the macrophages, to subside that inflammatory response, or not to let the inflammatory response or the protective response to happen. So in many cases, it is desired that the inflammation should be subsided, or there should be no inflammation. So this kind of, these two types of activations can happen in the case of macrophages. This one, the inflammatory or classical way, is called M1. In M1, activation is bactericidal, it's phagocytic and pro-inflammatory at the same time. And the cell secretes lots of interleukin 12, which would just enhance this process of inflammation. M1 is the dominating type, so mostly or most of the macrophages, whether these are monocyte-derived or others, would be activated, you know, uh, to like in one or the classical pathway. In a classical way, these would be activated, and when these macrophages are activated, they of course would be doing these jobs, which is bactericidal. They will kill bacteria, they will phagocytize different debris of the cells which are inside the body, and these cells would secrete cytokines, which would increase the inflammatory response inside our bodies. That those cytokines would, of course, be responsible for recruiting others, because the way or it's a proper language of the macrophages to talk to other cells. Like if it is going to invite other cells to the site of infection, it's going to secrete some kind of cytokines or chemokines in order to attract those cells to the site of infections. And so, because other cells also understand the language of these cytokines or the tissue or cellular hormones that we are talking about. Now, how this M1 or the classical, activation takes place. The classical activation takes place, when this cell or the macrophage comes in contact with some kind of a microbe. For instance, there are on the surface of the microbes, there are PAMPs, are pathogen-associated molecular patterns which are recognized by redundant receptors on the surface of the macrophages. These PAMPs actually are patterns. So a pattern, these are not highly specific, but these can be specific to groups of bacteria, groups of viruses, which are recognized the same way, and the macrophage then is going to deal with that groups of bacteria, groups of viruses or parasites in the same way. There would be no difference in the response, because this is cells of the innate immunity, and they are going to take up these microbes and process them, cut them into pieces with the help of different enzymes, or they have to activate different enzymes like NADH or other enzymes to produce reactive oxygen species or reactive nitrogen species that again are going to kill the bacterial species. So this is one way of activation of this inflammatory type or the classical pathway of the macrophages. For instance, LPS, lipopolysaccharide, which is common to, the wall membranes of several different types of, microbes. They have LPS, So all the microbes having LPS, they would be, this is a PAMP, LPS is a PAMP. So pathogen associated molecular pattern. So all the microbes which would have this LPS would be recognized the same way, and they would activate this macrophage in a classical manner or the inflammatory way. Similar is the case with damage-associated molecular pattern. DAMPs can also activate the inflammatory or the classical pathway. Others factors like some cytokines like the tumor necrosis factor and interferon gamma, for instance, interferon gamma and TNF can also trigger the classical pathway or the pro-inflammatory pathway. The pro-inflammatory pathway means that this particular macrophage is going to secrete cytokines that is which are actually responsible for promoting the inflammatory response. And uh, inflammatory cytokines and chemokines, which would be, released by these cells, which are going to affect other cells or attract other cells to the site of infection. Um, M1 macrophages utilize Warburg metabolism, and then two macrophages, utilize oxidative metabolism. So I would also tell you about what is the difference between Warburg metabolism and two oxidative metabolism in the next slide. But briefly, just I want to tell you here that this is a pro-inflammatory activation. M1 is a pro-inflammatory, activation, and M2 is, an anti-inflammatory activation. Two different types of activations of the same cell when it comes in contact with different stimuli. Some would be desired post-infection, some kind of activation like, for instance, alternative pathway, or if the infection uh, uh, has resolved, then this kind of activation, the anti-inflammatory, would be desired in order to make up for the damage which was done due to some infection or some injury or chemical injury or physical injury. While the inflammatory one, when there is some kind of infection, when a bacteria or virus or some other microbe enters into your body, then the cells, these macrophages, should be activated in a classical way, which is the dominant type, as I told you. In this, the anti-inflammatory or alternative pathway is triggered by some kind of cytokines like interleukin 13 and interleukin 4. Interleukins 13 and 4 are produced by T lymphocytes. Thus, T lymphocytes can also promote anti-inflammatory macrophage, activation. When the macrophage comes in contact with these cytokine has got a message. This is a message for, this particular macrophage to be activated in a different way, in such a way that this leads to the production of interleukin 10. So interleukin 10 actually is an anti-inflammatory cytokine. It is responsible for suppression of the inflammatory response. So whenever inside the body it is desired to suppress the inflammation, the cells actually are secreting interleukin 10, okay, that inhibit activity of T lymphocytes, natural killer cells, and macrophages. This interleukin 10 prevents excessive inflammation and damage to host cells. You would have seen in COVID-19 that excessive inflammatory response leads to death of the people. And people are using different steroidal options in order to cut that response, and because too much of the response can actually be very dangerous for the body. These alternately activated macrophages also produce growth factors that promote tissue repair. This response, these macrophages are producing different things like vascular endothelial growth factor, VEGF, which facilitates angiogenesis, and transforming growth factor beta, which facilitate deposition of extracellular matrix proteins for fibrosis. So fibrosis takes place, the extracellular matrix protein are produced by cells, in this fibrosis is needed. For instance, when there is some injury on the surface of your body, for a day you would see that there would be more infiltration of the neutrophils. The neutrophils would be there, and they would be fighting off that particular, necrotic tissue which is formed here and there, or some kind of the microbes that are going to enter into that infections, the wound. And after a day, that particular, a part of the cell, that particular injury site would be infiltrated by uh, macrophages. Initially, the macrophages formed there would be inflammatory, but later on, these are replaced by anti-inflammatory activation. The macrophages that are activated alternatively in order to subside that inflammatory response. And you would see there that there would be some kind of fibrosis at the site of infection, site injury, and that is required for just making up the damage which has been done by the wound. So that happens because of the deposition of extracellular matrix protein. The extracellular matrix is made of different types of proteins, which will be, for particularly fibrin, etc., would be deposited in that particular site. So this M2 is required. This type of activation, the alternative or anti-inflammatory activation, is required for wound healing or revascularization, which is desired some, post infection or injury, for instance. This type of phenotype, this type of activation will be required, or you would desire the macrophage to do to take part in this particular. So wound healing or a revascularization, some of the tissues which are damaged, the vessels are damaged, then these macrophages again, which are activated alternatively, can bring about this revascularization, making of the vessels, or re-epithelialization. If the epithelium of the tissue is lost, then re-epithelialization also takes place with the help of these macrophages which have been activated alternatively. So they have a role in uh, healing or repair. The macrophages that are, activated alternatively, or in other words, you can say in different processes related to development. The first one, the classical one, is related with some kind of infection or inflammation or something. So these are two different things done by the same macrophages and in different ways. Now, if we move on to the next slide, I just wanted to show you that how what is actually the Warburg effect here. The glucose normally, it is broken down into pyruvates, and the pyruvate again is either, it enters into the Krebs cycle, and this process actually, when the pyruvate is completely broken down, gives you 36 moles or 36 molecules of ATPs from a single glucose molecule that you get at the end, and some of the pyruvate is changed into lactate. But if there is no oxygen, for instance, then what would the cells do? When there is no oxygen, the glucose is converted into pyruvate, and the pyruvate again is converted into lactate. This is anaerobic glycolysis, which gives you only two molecules of ADP per molecule of glucose, or 2 moles of glucose or ATP per mole of glucose. So this is far less energy output. If you see here, just two molecules or 2 moles of ADPs is compared to oxidative phosphorylation, which gives you 36 molecules per glucose molecule. So the eukaryotic cells have adapted this kind of energy production after the oxygenation of the atmosphere billions of years ago, because this way is more efficient and it gives you more energy, and all the different functions which are going on inside your body require energy dependent processes, whether it is transport within the cell, or the cell's mobility or whatever the cell is doing, producing different types of proteins. All these are energy dependent processes. So if there is no oxygen, the cells would have been producing this much energy, which would not have been sufficient in most of the cases. Now, in some cases, for instance, in different types of tumors or proliferative tissues, where sometimes there would be deficiency of oxygen. Sometimes the oxygen is present there. Sometimes there is deficiency of oxygen. The glucose, when converted into pyruvate, mostly or in 85% of the cases goes into lactate formation because of the lack of oxygen, while only 5% goes into the Krebs cycle, which just gives you only 4 molecules of ADP, which is again not a very efficient process. This is called the Warburg metabolism or Warburg effect, which happens due to this insufficient oxygen which is there in the tissues. Now, if you look in the previous slide, as I told you, this type of phenotype, the anti-inflammatory or alternative phenotype of the macrophages is activated during, a tumor formation because you require a lot of different types of growth factors like VEGF, vascular endothelial growth factor, the tumor grows, and the new vessels formation is required for transportation of the nutrients and other things to the to that tumor mass. So that's why this type of Warburg metabolism would take place there, and this type of phenotype would be activated in the case of tumor cells. There are different functions of these different roles of the macrophages. They have a role in erythropoiesis, or the RBCs formation or iron recycling, because they would just take up the RBC and just break it down into pieces, and the iron which is formed is then recycled to the spleen for making use of it again for the purpose of cells making. Synapse pruning, the surfactant hemostasis, tissue regeneration, thermogenesis, insulin sensitivity. These are all different functions that these cells actually are doing. The pattern recognition receptors are there on the cell, receptors for apoptotic cells, because normally as I told you in this the very first slide, in normal conditions, when their apoptosis is going on inside the cells, inside the body, that is a continuous process, and that process should continue uh, inside the body without getting notice of any of the body, that something is going on inside the body. So that is a normal physiological response of the body to remove out the dead or diseased cells out of the body. So in those cases, the inflammatory type or the classical pathway, should not be activated in the case of macrophages, while the alternate pathway should be activated in that case, so as to subside any inflammatory outcome of the removing, the dead or diseased cells out of your system. So that was about the macrophage activation, and you can, if you are interested in PAMs and other things, you can find out the same material that I have already shared on those things. So thank you very much for your time, and hope to see you next time in another lecture. Thank you.